Hello, guys. I hope you can hear me. We will be waiting just a few more minutes just to see if more people are able to come in. And then we'll start straight away. So just a couple of minutes. Okay, I think we can start. First of all, I hope you can all hear me. I was able to give all of you the ability to unmute yourselves just to be able to promote questions. So if anyone does hear me, please let me know either on the chat. Yes, you can hear me. Perfect. Thank you very much. Okay, perfect. So let's start. You've just joined the introduction of pharmacogenomics, how do our genes affect how drugs work. I'm pretty much, the description of the lecture and a couple of learning outcomes. You can find those on the YUFE website. I'm sure you've seen those before you registered, so I'm just going to pass these through. I'm just going to tell you just a couple of things about myself, so you know who is talking. And my background is pharmacology. Pharmacology is the study of drugs. Basically, it's the science of studying drugs. I'm not a medical doctor. I'm not a clinician. I'm not a molecular biologist. I'm a pharmacologist. So I did study pharmacology in the UK. I did a couple of degrees connecting pharmacology and neuroscience. Neuroscience is the study of how the brain works, basically. I completed my training in the UK looking at opioids. Opioids are basically drugs, very strong analgesic drugs like morphine. I was trying to study how these drugs affect people's pain. I did a little bit of time in Italy at the University of Modena, where I continued studying the drugs against pain in various animal models. And I worked a couple of years in various pharmaceutical companies in Greece before I went back to the UK, and I did a little bit of teaching. So I started teaching high school kids and for a few years. I then started doing my own research at the University of Leicester, and then I moved to Australia at the University of Tasmania where I studied my first academic job as a lecturer in pharmacology. And I joined the University of Cyprus in 2015. I did quite a lot of work over the years in all these universities, but I'm just going to pass these couple of photos. all my work has been focused on how drugs work, right? And it's a huge content, right? You cannot really start giving introductions on pharmacology just with a couple of lectures, but this makes it a little bit harder for me and challenging for me today, because we actually have in this lecture people from 9 different countries registered in 18 different programs. So we do have people from studying law and business or medicine or biology to people doing literature. So people literally know nothing about drugs, nothing about how they work. And it's very challenging for me, I think, because the actual content of today is not just about what are drugs and how they work. It's a little bit more focused into a very futuristic part of drugs, which is called pharmacogenomics. It's basically the interaction between our drugs and our genes. So I'm gonna focus, I actually divided the lecture in 10 parts. I think it's going to take about an hour and a half maybe, and we're going to have a break as well in the middle. And I'm going to talk, first of all, the basics about the drugs, right? Because you guys don't really, most of you don't really know much about drugs. So I'm going to explain a little bit how drugs work and what are they and how do they do what they do. And then I'm going to start focusing a little bit on our genes. I'm going to talk about our genes, and then I'm going to talk about the interaction between the two. So bear with me, and we're going to have this nice bar at the top where I'm going to go through it, through the contents from 1 to 10. So every step of the way, you know roughly where I am in the lecture. If anyone has any questions, feel free to unmute yourselves and just ask a question, right? This is just, I want it to be as fun as possible for you guys. It's not a proper lecture with assessment and everything like that. So let's make it as fun as possible. If you have any questions, just unmute yourselves, just go through it and shoot your question. Right, so let's start. Before I start, I just want to tell you that I'm not going to be able to look at the chat box so often. So if you do have something of importance, please unmute yourselves and just interrupt me. That's fine. Okay, so the first thing is, what are drugs, right? That's the basic question. So drugs, we call them drugs, but basically we mean medicines, right? There's kind of a difference. Drugs is a little bit more of a general term, right? Everything that is doing something into our body is a drug, but not everything has an approval to be taken with a prescription or something, right? These are medicines. But for the sake of this lecture, I'm just going to keep the same term for both of them. So medicines or drugs are substances that we can reproduce, right? They don't fall from the sky. We can create them, right? And they have a very known chemical structure. Basically, we know what are they made of. And the result, if we take these substances, is that we have a biological effect in our body, right? Something happens in our bodies when we take these drugs. And we need to use them for therapeutic purposes, right? If a drug does something, but it's not really used for therapy, it's not used for a disease, then it's not a medicine, it's just a compound, it's just a substance that is doing something, right? And I'm going to give you a question, because it's not really straightforward of what is a drug and what is not drug. So, you know, bleach, right? We use it to clean stuff. Is bleach a drug? Well, it is a substance, right? It is a known chemical structure, right? We can produce it, right? And if I drink it, I will have a biological effect, right? I'm going to throw up, right? But it's not used for therapeutic purposes. So it's not a medicine. It's a chemical substance. Plant extract. So if I take a plant and I just boil it and take stuff out, right? Are these drugs? Are they medicines? And the reality is that for many, many plants, yes, they are medicines. And this is actually how we discovered drugs in the first place. We boiled plants and we took the stuff out and we drink it and then we use it for disease, right? So it fills all the definition of what a medicine is. Amphetamines, some of you might know, are illicit drugs, illegal drugs that some people abuse it for different purposes, ecstasy, etc. These are amphetamines. Are these medicines? Well, you'll be surprised, but they are, right? We do have amphetamines as prescription medicines, right? For specific condition. So amphetamines are drugs, are substances of non-chemical structure. If we take them, we do have a biological effect, and they are used for therapeutic purposes. Some of those amphetamines are used for therapeutic purposes. Vitamin K, right? Now, all vitamins we have in our minds that is something natural. We take them from fruits and vegetables, etc. Are these medicines? Yes, they are, right? It still fills that definition. There are substances, and we do take them for therapeutic purposes, right? If you have a deficiency of vitamin K and you take vitamin K, then you don't have the condition anymore. You are treated, right? So it can be a medicine. What about DNA, right? If I bite one of you and I get a flesh and I ingest it and I take your DNA and I eat it, is something happens? No, nothing happens, right? DNA, it is a substance. It is a chemical structure known. We can actually produce it in the lab. But if we do take it, it doesn't have any biological effect and certainly is not used for therapeutic purposes. Now, in the past, we tried to use DNA as a drug, right? We kind of used it, I'm not sure if you guys know, but the term is called gene therapy. Basically, you do get into your body some genes and they do something, and we try to use that for therapeutic purposes. It hasn't worked completely, but in the last decade, or actually less than that, in the last few years, we discovered new ways, new techniques in order to use DNA as therapy. So I'm pretty much sure that in the near future, we're going to use DNA as a drug. Caffeine, right? Coffee, basically. Coffee has caffeine among everything else. And caffeine is a known substance. It does have a biological effect. We drink it and we wake up, we're very active. Is it used for therapeutic purposes? No, it's not at the moment, right? Its effect is very, very weak, and we do have drugs that can do better job than caffeine. So it's not really used as a medicine, although it can be found in some pills as a substance, but it's not there to cure you for something. It's there to help the other drug that is in there to do a better job. So this is what the drugs are. And if we look at the human body, we can see that if we start from the atoms and the molecules and the stuff that makes us, and we go through it into a whole body, you will see different sciences looking at individual stuff, right? So biochemistry and organic chemistry is looking at molecules and how these connect with each other and what kind of properties they have. And then we move to something called organelles. So organelles are parts of cells, right? And the cells are obviously part of the tissue, right? And the tissue is part of the organ. So all these parts are being studied by cell biology. And then if we go to the organs and the systems, so the systems are basically organs coming together to form a bigger picture, then we do have sciences like physiology is looking at how these organs interact with each other, what do they secrete and how do they function. And then of course we go into the whole body, and that's where clinical medicine is looking at the humans and how all the systems of the humans interact with each other. And of course, the reason why I'm showing you this is the fact that the drugs are doing something into our body, but the question is, where do they do their job, right? Where do they act? And in order to understand that, we have to go as low as the cell level. So basically, let's look at a cell. This is a very, very high detail picture of a cell. And it's actually cut in the middle in order to see what's inside. Cells are just spherical bodies. And we can see the membrane of the cell. So the stuff that surrounds the cell is the membrane. And we see the nucleus, the center part of the cell. And this is where the DNA is, the genetic information. We're going to talk about the DNA and how the drug goes inside the cell and start interacting with the DNA. So apart from the DNA, which is basically all the information that create the cell, right? We do have other parts. And one of the most important parts of the cells are the proteins inside the cells. So these proteins, and I'm going to explain this a little bit later on, are actually coming from the genes. The genes create and give the instruction of how to create the proteins. But we see other stuff as well. We see a strange thing called Golgi and something called endoplasmic reticulum. So these structures in the cell are basically structures that help the cell transfer the proteins through the cell into the cell surface or inside the cell to do stuff, right? The proteins don't just swim inside the cell, they are transferred. In order to transfer them, you need all these structures. And we need vesicles. Vesicles are basically just sacs of proteins inside and different chemicals. And we need vesicles in order to tidy the cell, right? To organize the cell. We can't just have things floating around. We need it to be organized. And we have other organelles like mitochondria. So mitochondria are specific organelles that produce energy, right? So the cell is a factory of proteins. It produces proteins and it needs energy. So mitochondria take stuff from the environment like oxygen and water, etc., and they produce energy. And they use this energy to do all the functions of the cell. So this is roughly, very, very basically what the cells are. And we have some interesting numbers about the cells. Actually, your body has 37.2 trillion cells, right? It's a huge amount. But if you take one cell and you take the DNA of the cell and you stretch it out, you reach about 2 meters, right? It's very, very tiny, but it's very, very packed into a very, very small place. And it's unbelievable how long the DNA of one cell is. And if you look at the proteins of the cells, and this is because I told you before, the proteins are the very, very key ingredient of the cells. We have 42 million proteins per one cell, you know, and we have all these 37 trillion cells. So we'll talk about a vast amount of proteins in our bodies. So what are all these guys are doing? So this is a graph, a figure, and it shows you a depiction of a cell. So you do have the nucleus, and inside the nucleus, as you see, is the DNA. And you do have all these Golgi and the apparatuses that they create the proteins. And you see all these things are searching and creating stuff that are called receptors. And we put them in the cell surface. And these receptors are actually proteins that receive signals from the surroundings, right? And this is what the cell is doing. The cell is always reading the surrounding to see where am I, right? What kind of signals do I have? Is the temperature below the proper limit? Is the pH below? Do I have mediators and chemicals from the surroundings to transfer messages? So the cell needs these receptors in the cell surface. And all these, as you see here, they're created from the DNA. So the DNA is giving the instructions of how to create these proteins, and it produces these proteins that reach the surface. But sometimes things go wrong, right? So if you see here, this structure called the ribosome is important to translate the DNA and the information into protein, but sometimes it's not doing a very good job. So it creates all these abnormal proteins that don't really go to the surface. They just accumulate in the cell. So the cell actually has what we call a pathology. Is something wrong with it, right? It creates disease. And this cell eventually dies, but sometimes it doesn't die and it creates symptoms. And this is why we do have the effects that we have in some cells. So if you really look it up from zooming out, you see that the whole purpose of the cell is to produce proteins, right? And it does that through different structures like DNA, but the DNA is not really translated straight away to proteins. It goes through something else that is called RNA, and actually is called messenger RNA. And you guys probably have heard this word before, because we do have mRNA vaccines for COVID, and we're starting introducing this word into the general public because of vaccines. But actually, we all have mRNAs in our bodies, and mRNAs are created in the nucleus from the cell, and then they are going outside of the nucleus, and they go to the ribosomes, which is actually other proteins, in order to create all these end receptors. So in this kind of picture of a cell, what is a drug doing? All right, what's the aim of a drug? The aim of the drug is actually a triple aim. The first aim is to correct a problem. So over here, if you see clearly, you see some proteins that have been misfolded. Their shape is not right? This is the actual shape of the correct protein, of this specific protein. So this is something else. It's an abnormal protein, right? So we can use a drug to correct this mistake and to delete these abnormal proteins. Another aim of the drug is to counteract a problem. Counteraction of a problem is not fixing a problem. It's just giving you help. It's helping cells to overcome the problem without actually fixing it, right? So let's say that you have a blow tire in your car, and your tire goes poof, and it has a hole, right? But if I take the tire out and I'm putting a new tire inside, right, that's fixing the problem. But if I patch the broken tire, then that's not really a fixation, that's a counteraction, right? So we have drugs that can do this. And we also have drugs to protect you from a problem, right? So you might take drugs because you want to defend yourself from creating a problem, right? So this is the aims of the drugs. But if you look at the fact that the cells have many proteins, and actually most of the cells are proteins, the target of the drugs are proteins as well. So the drug is a chemical substance and is going to do something into the cell, and it needs a target, right? It's like a missile with a target. So this target is usually proteins. So let me give you an example. So let's say that your father has high blood pressure, right? And this is called in the clinic hypertension. So basically your body has blood vessels that they carry the blood, right? And these blood vessels are, they have pressure inside because it's a liquid, the blood is liquid. So the heart is pumping out blood in the periphery, in your body, and then it takes the blood back to the heart, and then it takes it to the lungs because you need oxygen in the blood to transfer it into your body, right? So this is what the circulatory system, the cardiovascular system is doing. But all this is in liquid form, right? The blood is in liquid, so it has pressure. Now, if my pressure is higher than normal, then I have hypertension. I have increased blood pressure. And one cause of hypertension is the fact that our blood vessels are constricted. Their diameter is less than normal. And because the blood is liquid, if the diameter is small, then there is high pressure inside the system. So what can we do for hypertension? Well, one thing that we can do is to dilate the blood vessels, right? If you dilate your arteries, then you leave enough space for the blood to do that. And then you don't have a hypertension anymore, right? So the question is, how do I do that with a drug, right? So if you look closely in your blood vessels, you see these red circles. These are muscles. So there's a muscle inside your vessel that constricts and dilates. And this muscle is actually what can create the hypertension and what can create the dilation. So if I go to a muscle cell and I look closely in a muscle cell, please ignore the details of the chemicals that you see here, but the focus is the fact that there is something called calcium. So basically our normal calcium, what you take from cheese and milk, right? This calcium gets inside the cell of the muscle, and it does that through channels. And once it goes inside, the cell contracts, the muscle cell contracts, it causes a contraction. And if your blood vessels take a lot of calcium, then they contract, right? So obviously, one way to give a therapy to a person that has high blood pressure is to stop this from happening, right? So what's the solution? Well, the solution can be to give a drug that can go to this channel and can block this channel and doesn't let calcium go inside. So it doesn't cause contraction and it causes relaxation. And this is what we actually do in the clinic, right? So we have this drug that goes to this channel and it just blocks it. It just sits on top of it and doesn't let calcium get in. So if calcium doesn't get in, you don't have calcium inside your cells and you don't have a contraction, and therefore your blood vessels dilate, and therefore you have a relaxation and you don't have hypertension anymore, right? So as you can see, a human being has a disease and we try to figure out the particulars. What's the mechanism of this disease? Once I found the mechanism, I have to find a target. A target is usually a protein. In this situation, a target is a channel. So if I give a drug that blocks this channel, then I have a solution, I have a therapy. And the question now is, what kind of targets? What else can I target, right? What else can I use drugs for? And we have different types of targets. So this is just an example of a cell. And on top of this cell, you have different types of targets. You see these 4 targets. The one target is a channel, is the one that I just explained. So basically ions like calcium and magnesium and sodium and potassium, all these different ions, they go inside the cell and is doing something into the cell. And I can block this something that is doing. The second type of targets are just proteins that are sitting on top of this cell. And once I give it a drug to do something, then it sends a message to other proteins in the cell. And once it propagates a message, then I have some kind of an effect inside the cell. So this is not a channel. This is just a protein that receives a drug. This is why we call it a receptor. And we have other receptors that are not so complicated as the middle ones, the number 2 ones. And these are different types of receptors. We call them enzyme-linked receptors. Is doing different stuff inside the cell, but they're doing it themselves. And we have the 4th type of receptors that are not really in the cell surface, they are inside the nucleus, right? So they're very close to where the DNA is. And if I have to give a drug to target that kind of protein, the drug has to go inside the cell, inside the nucleus, find the target, the receptor, and then once he attacks this target, is doing something to the DNA. And then this something creates an effect on the cell. So at the bottom of this graph, it just gives you some time scales. And what this time scales is actually how long does it take for a drug to be effective based on what kind of target you're targeting. And you can see here, the drugs are very, very fast. Actually, there's seconds in terms of their effects, are the ones that are targeting ion channels. If your drug goes inside the nucleus to target DNA, then it takes a lot of hours for the drug to have an effect. And I'll give you an example, right? So nicotine is a drug, right? We don't really use it for therapy, so you can't really call it a medicine. I mean, in some very, very rare cases we do, but not usually. But you do have it in cigarettes, right? So once you get a cigarette, the people who smoke might understand, Once you take this first puff, straight away in seconds, you have an effect in your brain, right? You can feel the nicotine having an effect. And the question is why is it so fast? It's because nicotine goes and attacks an ion channel, right? It attacks an ion channel, and then this ion channel has a very, very fast effect, and that's why you feel the effect very fast. So I'm very sorry to give you a little bit in-depth biology to people that don't really have a background, but I just wanted to stress out the complexity of the drugs and the targets. And the targets are very complex, right? The drugs are not that complex, it's just chemicals, but the targets are very complicated. So the question now is, okay, you have all these different proteins and all these different targets, and you have drugs, right? Chemicals. So these two come together, and you say that they interact. So what do I mean by interaction? The interaction is exactly like a lock and a key. And if the lock is the receptor, the protein, and if the key is your drug, then the key fits in a lock, right? This is exactly what happens in drugs. Drugs fit inside the receptor, but they do so in a way that their shape is important. Not every drug can bind every receptor, every protein. This interaction is very specific. It's very specific because it requires a fitness. It requires the drug to fit inside the receptor, exactly like the key and the lock. So if I give you nicotine as an example that I gave you before, it will go only to the receptors that fits very nicely to nicotine. And these are called actually nicotinic receptors. They only accept nicotine, right? Why? Because the shape of the receptor is such that only nicotine can actually bind into it and fits perfectly into it. And this actually means, if you think about it, that not every drug is doing many things. Drugs are doing very specific things because they have a shape and because they can affect only the proteins that fit into that shape, right? So this is actually a very good way of explaining why, if I take a drug, right? What about if I have some effects long term, right? What about if I do something and I don't know what this is doing, what the drug is doing? Well, actually, we can very much predict what the drug can do because we know the shape of the drug, and we actually know nearly every protein in your body. We know their shape, right? So we can run simulations in computers, and we can do all this screening, right? And we can see if I take this drug based on its shape, where does it go? What does it do? And I can predict what the drug can do. So it's not really a magic. It's not really very agnostic. It's not really unknown of what the drug can do. Apart from the fact that before I give it to humans, we test it, right? So we actually confirm what the screening is doing. But this gives you an idea of the fact that every drug that we have and every drug that we can invent in the future, we will be able to know in a very, very big percentage of possibility what it will be able to do. But if you see a little bit in reality, it's not really a key and a lock. It's a little bit more complex, obviously. And this gives you a rough idea of a simulation in a computer, right? On the left-hand side, you can see the receptor. And on the right-hand side, you can see this strange bulky molecule called the drug. And you can see that it fits nicely into the receptor, but you appreciate the 3D shape of the things, right? So when I said before that the lock and the key, they have to have the right shape, I mean the three-dimensional shape, right? So the 3 dimensions are very important. So binding a drug with the receptor, what happens then? If you look closely to this simulation, once the drug goes inside to the receptor, there's some sort of a movement, right? There is these little blue and red things on the receptor. These are actually amino acids of the protein. And you see that they move when the drug goes inside, they kind of move a little bit. This is what happens in reality. The receptor is changing its shape a little bit. And because it does change a little bit its shape, then this change is important in order to create this biological response of the cell. So if I take a drug and this drug goes to a receptor and the receptor changes the shape, that change will create a response of the cell, and the cell might, for example, contract if it's a muscle, or it secretes something if it's a gland, right? So different parts of our bodies, different cells, different functions. But this is a very key part. It creates a movement, and the movement is important for the effect. And I'll give you now another example. There is a very known drug called morphine, right? Morphine is actually a medicine. You use it to relieve you from pain, very, very intense pain. You can't just take it for headache. You take it for very intense pain. For example, cancer pain is a very, very intense pain. Only with morphine goes away, right? And you take morphine, and we call this analgesia. Analgesia, basically, it's like the pain goes away, pain-killing effect. And what you see here is you see neurons, right? Nerves. So the nerves are the cells which are important to carry out the signals. And if you think about it, pain is a signal, right? If I touch something sharp and I create some kind of a pressure in my fingertips, there is nerves there, and they will create a signal. They will go to my brain, and it will tell my brain, oh, this is very painful, it's very sharp, take your hand away, right? This information, which I then translate into a pain signal in my body, is governed by cells. And if you look at the left-hand side, again, calcium is very important for this signal. So calcium goes inside the cell, and you have this flash of the nerve signal. This is the pain signal. It goes to my brain, and I feel pain. But I also have some proteins on these cells called opioid receptors. Basically, are receptors, proteins, but they're called opioid because opioids are all the drugs that are like morphine, for example. Morphine is an opioid drug, right? It comes from opium, right? So if I take morphine and I send it into the nerve, it goes and it blocks, it attacks actually this receptor, and it changes the form of the receptor, and that sends a signal inside the cell, and the signal is stop the calcium from getting in and open up potassium to go out. This is to people that do not have a background in biology, these might sound like strange. Okay, you stop calcium going in and you throw potassium out. Why do I do that? Well, that actually is a signal for the cell to stop transmitting. You see here the transmission of the pain? You don't see it here. So basically, if I throw morphine in my cells, if I take it, it will go to the opioid receptors and it will tell my cells to stop getting calcium inside, and that will stop the pain signal. So basically, this means that the calcium stopping from entering my cells will cause an inactivation of the pain signal. And actually, this is called analgesia. So as you can see, here, I don't actually block something from going in by using morphine. If you remember previously, when I talked about the calcium and the muscle cell and the hypertension, I had to block the ion channel, right? Over here, I don't really go to the calcium channel, I go somewhere else, right? So these are two different types of properties of drugs. They can block stuff, but they can actually do stuff. And this is why we divide drugs into two categories. We divide them into agonists and antagonists. Agonist is a drug that is doing something actively. And antagonist is a drug when it blocks something, right? So by using these two definitions, you can understand that the first example which I talked about, the hypertension and the shrinkage of my blood vessels and the blocking of the calcium channel, this is an antagonist drug. Agonist drug is morphine. Morphine goes to the receptor opioids and it sends a signal into the cell. It doesn't block anything, right? That signal creates a stoppage of calcium. So it's an indirect stoppage of calcium, but the actual drug itself, it creates a signal. And that's why we call these agonists. Every drug in the market is either an antagonist or an agonist. They both do stuff, they both create an effect, but they are doing it through a different pathway, a different kind of property. Okay, so in order to discuss how drugs work, we said that it goes to proteins and is doing stuff, and it goes through specific receptors, and it requires appropriate shape, three-dimensional shape to do something into the cell. But what about the dose of the drug, right? So let's say that I give you a little bit of morphine, or I give to other people a lot of morphine, right? These two people will have different effects, right? It's not going to be exactly the same effect of this drug to the person. So why the dose plays a role? Well, I have to take you all the way back to the 1400s, right? Paracelsus is considered the father of pharmacology, or the father of medicinal chemistry. And this person actually, by studying toxins and poisons, that it says all things are poison. I'm trying to do the voice, right, of Paracelsus in 1400 and 1500 something, right? Let's try. All things are poison and nothing is without poison. Only the dose permits something not to be poisonous, right? Something like that. But what does this mean, right? So he says everything is poison and nothing is poison. It doesn't really make sense, right? Actually, it does make sense, because if you think about it, let's say I give you this calcium drug when you have hypertension, right? And I give you this a calcium channel blocker, right? But I give you just a little bit, like just on the fingertip, on the fingernail of this little tiny substance. It won't do anything to you. Why? You took your drug, right? Why doesn't it do something? And the opposite, if you take a little bit of nicotine, very, very tiny little bit amount, it won't do anything to you. If you make it the other way around, if I have something which is very natural, let's say water, and if I drink a lot of water, well, you actually die, because a lot of water will dilute your blood so much that you won't have high concentrations of sodium. Sodium is the #1 ion that is important for your brain to work, and you will go to coma, right? If you take anything which is natural and safe into a very big amount, it will be poisonous, right? So Paracelsus really hit the nail on the head right there by saying that anything is poison and nothing is poison. The dose is what calls something a poison and not a poison. So the actual dose of the drug is the ultimate importance of the amount of the effect that you will have. And this is actually the foundation of pharmacology. Actually, by looking at if the dose has any effect, you can actually understand if this is a drug or not, right? I'll give you an example. You all know what placebo might be, right? Placebo basically is just an effect that you have, but it's not because of the drug, it's because of your psychology or your imagination or whatever. Your brain is powerful enough to create changes in yourself just by thinking something, right? It's not very high tech, it's not very wow, it's very natural. If now I'm thinking about pizza and I'm going to start creating secretions in my stomach, right? I'm going to start being hungry just by the thought, right? It's exactly the same for drugs. If you give me sugar and you tell me, oh, actually, this is a drug, you're going to feel fine, right? After taking this, I might feel fine, right? But I didn't take drugs. That's a placebo, right? So if I take a placebo, if you give me sugar and you lie to me and you say that this is a drug, right? If you give me more sugar and more sugar, I won't have more and more effect. I will have the same effect. The placebo has only the same effect. It doesn't fluctuate based on the dose, obviously because sugar doesn't do nothing. This is a way to differentiate placebo from an actual drug. An actual drug, if you increase the dose, you have to see increased effect. A placebo, if you increase the dose of a sugar, you won't see increased effect. Actually, this is the actual test that we use in the clinic to understand whether the effect that we had from our patient was because of the drug or whether it was from just the other processes, the cognitive processes of the brain. Okay, so I'm going to finish talking about how drugs work by zooming out and by saying all these three things. These three properties is the properties of all drugs. Drugs have a binding force to their target. This is called affinity. They bind with their receptor. It's very important. If your drug doesn't bind to the receptor that you want, it won't have the effect that you want. The second thing is the biological effect of the cells. This is called efficacy. So your drug might bind to the receptor, but might not do a good job, right? So actually, efficacy is the way to compare different drugs with each other. And then we have what we call potency. Potency is basically the strength of the drug. The strength of the drug has to do with the dose. And I'll give you a small example. Let's say that I have hypertension, and a doctor says, okay, I have two drugs that can cure your hypertension. One drug, it will cure it with 1 milligram. The other drug will cure it with 10 milligrams, right? Obviously, this means that the drug with the 2 milligrams is a stronger drug, right? It will treat me similarly to the other drug, but with a very small dose. That makes it a strong drug. We call this potency. We actually like drugs to be potent because we don't want to give a lot of drug to the patient to have the effect, because a lot of drug means that you might have side effects, more risk of side effects than a little bit of drug. So these three properties are the properties of every drug. So now that we talked about how the drug works in our body, let's talk a little bit the other way around. What does our body do to the drug? And this is actually, maybe it may sound like a strange question to someone, because they say, okay, what do you mean? The drug goes inside and it's doing something. What do you mean my body is doing something to the drug? Well, if you think about it, you take your pill, right? Where does it go? Well, it goes to my stomach, okay. And where does it go after that? Well, it goes to my targets. Well, your targets might be to a specific organ, right? For example, I said before, hypertension, your target is the muscle cells of your blood vessels, right? So how much drug does it go there? All of it? No, because you have what's called a metabolism. You have your kidney, your liver to metabolize your drug. You have your kidney, which is the organ that creates urine to excrete the drug, right? To take it out. Actually, your drug can be excreted through the sweat, right? So not all the drug goes to the target. This is called pharmacokinetics. Basically, it's a study of where your drug goes once you take it. And the pharmacokinetics discusses these four things: absorption, which is how the drug goes in and how does it absorb. Distribution is where does it go. Metabolism is how does it change by my liver, basically. And then excretion, how much of it, how fast does it go away, right? So I'll give you an example. If you go tonight to a pub and you get a drink and you drink alcohol, alcohol is kind of a drug, right? So it's a chemical, it's not medicine, it's a drug. It goes inside, right? If you take alcohol and you take like a glass of alcohol or 2 glasses of alcohol and you start feeling dizzy, right? It has an effect, right? It's because it went to your brain. It didn't go to your toes, right? It didn't go to your fingernails. It went to the brain. It went to the stomach. You might feel a little bit sick to your stomach. it goes to specific places. It doesn't go everywhere, right? So that's distribution, right? And then you feel dizzy and you say, oh, I had a little bit too much. I have to wait a little bit. If I wait, then after a while, it will go away. I'm going to feel better. Why? Right? Because your drug, the alcohol, it will start decomposing because of the metabolism. And then you go to the toilet a couple of times and you take the drug outside of your system and then you feel better, right? So the pharmacokinetics is very important into what my body is doing to the actual drug, right? And for how long. So talking about absorption, the alpha into the ADME is all these little colors, all these spherical things are, let's say, are drugs, and they have to go through your cells somehow, right? And they go through your cells by different ways. They are absorbed by your cells. Now, if you think about it, if the drug that I take is a very big molecule, a very big structure, it won't be absorbed easily, right? If it's a very small molecule, it will be absorbed easier. So actually, the properties of the drug, the size of the drug, it will define how much will go into my system. Also, the way you take the drug affects what it does, right? So imagine you don't drink alcohol. Imagine you just rub it, right? You take, let's say you take like a beer and you just throw it on your skin, you rub it. Do you get alcohol inside your system? No. Why? Well, you're actually going to get a little bit of alcohol absorbed through your skin, but that's going to be a very tiny amount because I have many, many cells that the alcohol won't be able to go through those cells. So it won't be absorbed in my circulation and it won't go to the brain. Now, let's say that I have a drug that I want to take for pain, right? And I take this drug through the nose, right? Let's say that I sniff this drug instead of just taking it through my stomach. Will it work better for pain? Yes, it will work better for pain. Why? Because your nose is very close to your brain, right? Once you take your drug through the nose, it will be absorbed through the circulation, but the brain is very close and it will get into the brain faster, right? So it will get faster. Whereas if you take it as a pill, it will go to your stomach and it will take a lot of time to go distribute and then go to the brain. So if you see, the way that you take the drug is very important into the effect of the drug. This might be a little bit complicated. It's not that complicated, but it actually shows you all the different organs, right? You see the muscles, I have the heart here, I have the liver, and then you see I take the drug, and where does the drug go, right? All this thing here, the red thing is my blood vessel, it's my blood, right? So if I do an injection straight away into the blood, that's faster, right? Because it will go, it will distribute itself in all my body very fast. If I take a drug through a pill like here, it will have to go through my stomach, it will have to go through the circulation, it will be metabolized by the liver, then it will go to the circulation, right? It takes a long time. If I take an injection into the muscle, that's called an intramuscular injection, actually that goes faster than the oral injection, sorry, the oral pill, but it's slower than the intravenous injection. So you have all these different ways of getting drugs inside your body. The way you take it inside, it will define not only how fast it will act, but it will only decide where it will be distributed into your body. And of course, if I have something like an eye condition, let's say I have infection in my eye, it's faster if I take a drug through a drop in my eye rather than taking it through a pill. Now let's go to the me of the ADME pharmacokinetics. Me stands for metabolism, right? And when we talk about metabolism, this is very important actually. It's the liver. The liver is metabolizing the drugs. What does it mean? It means that it changes the drugs, right? So the liver is not doing just the metabolism. It does a lot of stuff in our body. It's a very, very important organ in our bodies. It creates amino acids, right? it absorbs vitamins from food, it creates cholesterol, and it's very important. Cholesterol is very important because it creates all these hormones, testosterone and estrogen. All these are created by cholesterol. And it regulates glucose in your system, right? Very important jobs. But it also metabolizes drugs. So this is here, deactivation of poisons and toxins, right? This is the stuff that is doing to your drugs once you take it, because let's face it, anything that you take inside, your organism will understand that it's something from outside, right? So it will behave as if it's something dangerous. So it will try to deactivate the drug. And this deactivation is called biotransformation. Basically, it transforms the drug into something else, right? What's this something else? Well, we call them metabolites. This is the drug. The drug is the stuff that I take inside. And then my enzymes in the liver is doing something to the drug, and then it comes out as metabolite. It's not really coming out as a drug. It's coming out as something different, right? So the focus here is on these enzymes. These enzymes, which are called CYP450 enzymes, these are the enzymes of your liver. They destroy the drugs, right? And I have different forms of these enzymes. I'm not going to bother you with too much numbers. I have many, many different families. And every enzyme has a code, like this one, CYP2E1, okay? But every little aspect of this name, it gives you an idea of the family of the enzyme, of the subfamily of the enzyme, and the family and the form of the enzyme, right? And this is just an example of aspirin. So aspirin has this type of chemical form. Once I take it inside, my liver is doing this. It produces glucuronide, right? It's a different strange chemical. Basically, it's not aspirin anymore. So it won't be effective. Glucuronide doesn't do nothing for pain. Aspirin does, right? So that's a problem, right? In drugs. Now, if you have pain and I give you aspirin, you don't want your liver to destroy aspirin, right? Because you want aspirin to relieve you from pain. But liver won't ask you, right? The liver will destroy aspirin whether you want it or not. So what do? Basically, well, you say, look, these are enzymes, right, that are destroying the drug. Well, enzymes are destroying it, but they can't destroy everything. But like they are doing some work. So I'm going to give you, the human, more aspirin than the enzyme can destroy, right? So the enzyme will try to destroy all aspirin. It won't be able to do it all because it's going to be a very big amount. So the amount that won't be able to destroy, this amount will be effective, and it will go to the target, and we will reduce your pain, right? So before we give the drug to a person, we know what the liver is doing to the drug, and we change the dose of the drug in such a way so we are left with some drug behind after the liver gets done. And of course, you see here something very interesting. You see where your liver is, right? Your liver is very high into your abdominal cavity. It's very close to your stomach. Why? Because your body wants to destroy anything that you take from your mouth, right? This is the initial route of administration of food, for example, or ingestion of some poison or something, right? Now, if I give you the poison from your vein, it won't be destroyed by the liver, right? It will go straight to the blood. And if it goes straight to the blood, it will go straight into the target, and then it will do the job, right? And if you think about it, this is why people that are abusing morphine and opioids and narcotics, they are doing injections, intravenous injections, because they don't want the expensive morphine to be destroyed by my liver. So they do it by injection, and it goes straight away into their brain, and it has an effect, euphoria, etc. That's why these guys abuse these drugs. So the liver is getting everything that goes to your stomach and your intestines. It absorbs it. it destroys as much as possible, and then the rest goes to your circulation, right? So it does this by the CYP enzymes, as we said, but look at this here. You take 100% of the dose, basically all the drug that you want. It goes through your stomach. Okay, fine. It's still 100%. Then some of it is absorbed from the liver, 70%. Where is the next 30%? Well, the next 30% is either absorbed in my bloodstream through the intestine, or I just create feces or something else, right? So 70% is absorbed by the liver, and only 15% can go through the liver without being metabolized and goes to my circulation. So in this example, you have these 15% that definitely goes into the bloodstream, and then you have another 30% that goes somewhere else, right? So the dose that you give in the beginning, if I take it as a pill, only 15% will go to my target. All right, okay, forget about this. I give you the names of many, many drugs, not to confuse you, just to give you an idea that one enzyme is not doing any metabolism just by one drug. It metabolizes different drugs, right? So the CYP3A4 enzyme that you see here, it metabolizes all these drugs. The CYP2C9 or 8 enzyme, it metabolizes these drugs, right? And the reason why I'm showing you this is because if you take two drugs, let's say that you have two conditions, right? Let's say that your grandpa or your grandma has high blood pressure, but it also has, let's say, an infection, right? So your grandpa will take an antihypertensive pill for the blood pressure, but it will take also an antibiotic. If these two drugs happen to go to the same enzyme, then that's a problem, right? That's a problem because your enzyme in your liver won't be able to do the job for both drugs. That means that some of the drug that your grandpa will take will go unmetabolized into your bloodstream, which means that it will have more effect than you want to, and then more effect means more side effects as well, right? So when a doctor subscribes different drugs to the same person, it makes it sure that these drugs won't interact with each other when they go to the liver, right? And if they do, then it gives you another drug. It changes the drug to something else. And of course, apart from these CYP enzymes, we have other enzymes as well. I'm not going to bother you with these, that they also do metabolism. Okay, and let's finalize with excretion, right? The excretion. The excretion is basically your kidneys. Your kidneys are a little bit higher above your abdominal, but it's at the back of your cavity, and the kidney is actually filtering the blood. It's filtering the blood with all the substances that the blood has that maybe your body doesn't need anymore, and it's filtering it out into a urine, right? You throw it away, right? And it's not, obviously, it doesn't filter it 100%. So it does need some time to filter a compound, a drug, a medicine out of your body, but eventually it goes out, right? So eventually all of your drug, all the drug that you take, eventually it will go outside if you don't take more, right? So if you take one pill, you will have the absorption, you will have the metabolism, you will have the distribution, the drug will go to your body, you will have the effect, right? And then if you wait a few hours or days, maybe, then the drug will be excreted because it will keep your kidneys will keep filtering the drug outside of your body. So this is, I don't know why this doesn't go away. Anyway, this is the way that the drugs work and how my body affects the drugs. So we have the targets and we have what our body is doing. And now I'm going to start talking a little bit about genes. And I'm going to talk a little bit about genes, then I'm going to pause. So I'm going to pause in a few minutes, just hang in there. and then we're going to pause, and then we're going to talk a little bit more about how the drugs and the genes come together. So talking a little bit about the genes. Obviously, when I talk about the genes, everyone knows that we talk about DNA, basically, right? These are the genes is in the DNA. So this is the cells, and in the cells we have the nucleus, and inside the nucleus we have the chromosomes, right? And the chromosomes are basically just structures that have packed DNA. If I unpack this string of DNA, I can see this double helix, right? The DNA is a double helix. But if I go a little bit more into the DNA, I can see all these strange letters, right? I see T, A, C, G. These are called base pairs. Basically, what these are 4 substances, 4 compounds, 4 chemicals that are pairing together. That's why we call them base pairs. A and T, G and C. Right? Basically, these are called adenine, thymine, guanine, cytosine. These are the initials of the compounds, but they form pairs, right? And this is what the DNA is basically. It's different pairs and the combination of different pairs together. So this is the double helix, and it has all these combinations. A goes always with T, and C goes always with G, but the sequence of one another can be mixed. This is our DNA, right? This is our DNA. And obviously, to the guys that don't know about DNA and they have other focus, they obviously they don't understand nothing. They just see letters, right? Random letters. Well, they're not really random, because if I divide them into threes, right? Look at these, 3s and 3 and 3 and 3. These are called codons. These 3 will actually be changed, transcribed by my cells into RNA. And the RNA is just basically one strand of the DNA. And then this will be translated into an amino acid. So every codon is one amino acid. And this one amino acid, if I stack them all together, I create one protein, right? Remember, I told you that the genes are creating proteins. That's the whole purpose of the cell, right? And this is how they do it. You have all these C, G, A, T, C, G, A, T. These, if you cut them in threes, every 3 pairs, they create one amino acid, and every hundreds of amino acids, they create one protein. And the question is, this is why it's complicated, because the different combinations of C, G, A, etc., they create different amino acids. And of course, I don't want to give you too much detail, but I'm just going to show you here just an idea to see how complicated it is. This is the first position, this is the second position, and this is the third position of the codon. And you see these 1, 2, 3, they will create a specific amino acid. All these are the amino acids that we have for proteins. So basically, pro is the short name for proline, is one amino acid. And you can create proline by having a CCA. CCA gives you a proline, fine, right? So if I have here CCA, it will give me a proline. A CCG will also give me a proline, right? So the organism, my cells, are reading these DNA and they're creating proteins. That's all they do. So from DNA, I create RNA, and from RNA, I create protein. That's what my cells are doing. That's why the genes are there. That's why the proteins are there. And this is what life is all about. My genes, your genes, are creating constantly RNA copies. These RNA copies create constantly proteins. And these proteins are actually creating life, right? You have functions. You have all these jobs that your organs are doing. Your cardiac muscle is contracting and is beating, right? This is muscle cells that need to constrict, and it is proteins to do that, right? So life is based on DNA, RNA, and genes. And all this is based on how this little code here translates into proteins. We have 6 billion pairs in our DNA, and we have 40,000 genes into our DNA. Now, the question is, why do I have less genes than pairs? And that's a very critical question. Is because for some reason we have base pairs that don't produce proteins, excuse me. They're just there. We don't know what it's doing in our system. It's called silent base pairs, silent genes, basically silent DNA. It's not creating proteins. It's just millions and millions of base pairs of nothingness, right? Right. In the beginning, we thought it's just empty. It's not doing anything. We think now it's part of our evolution. It used to do something that it's not useful anymore, and it stopped doing it, right? So, and we don't need to go into too much details here, but all I'm going to say before I stop for a break is that this codon here might be different from one person to another. I might have a mutation to a specific codon, to a specific pair, and this is called single nucleotide polymorphism, SNP into a short. Single, one change. Nucleotide is actually the base pair into biological language. It's called nucleotide. Polymorphism is a fancy word for mutation, right? So basically a single mutation in a single base pair, it will create a different protein. So if this code here needs to be creating a specific protein, let's say a calcium ion channel, right, in your cells, if me as Nicholas has one specific mutation, don't ask me why I have this mutation. Maybe it's through inheritance. Maybe my mother had this mutation and her mother and her mother, or maybe I had it afterwards. Basically, I had a toxin that I ingested a long time ago and created a mutation. We can have this kind of effect, right? Nevertheless, this mutation will create a different protein. And this guy is the source of why we're all different. We're all different because we all have different mutations. In every body that you see in the population, we're all different because we all have mutations. Nothing is similar from one person to another. Nothing in totality, right? We have proteins that are similar, obviously, because we're all humans. We all have a heart that beats, right? So we do have proteins that are similar, but we have many, 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 many changes and differences. So my genes are different than yours. If my genes are different than yours, it means that I have different proteins than yours. And think about it. What does this mean in terms of drugs? If the proteins are different, the drugs have different effects. What about the genes? The genes are different. Of course, the targets of the genes, the target of the drug, are different, right? So all the stuff that we talked about the drugs can be different from one person to the other. And this is why if I take a drug and you take a drug and it's exactly the same drug, it doesn't mean they will have the same effect. And I'm going to stop it here as I'm going to make a break. I don't know if you guys have any questions that need answering at the moment. We're going to have a nice break of, let's say 10 minutes, let's say 10-15 minutes roughly. Let's make it 15 minutes just to make sure that everyone is nice and rested, stretch their legs and hands, have a sip of water, and then come back in 15 minutes exactly to continue and talk now a little bit about how these two interact with each other, how drugs and genes interact with each other. Okay, I'm going to pause. Thank you very much.
Hey, is it you, right?
Yes, it is. Highlighting? Yeah, but that's fine. That's completely fine.
Just to know, intervention
Okay.
exam, you have remove the thing.
Yeah, obviously.
You can put it in crayon or something. Then if you see something written on it, remove it so that you don't.
Yeah.
There's nothing about anything on it.
Okay, so just post it then.
That's the whole big book you will study. Good luck with this.
Thank you so much. I'll return it back to you after the exam.
Yeah.
We'll have it back.
Yeah, you tell me when.
Yeah. Great. Thank you so much.
No worries. Goodbye.
Okay, we're back. Sorry. Okay, so until now, we just talked about the basic pharmacology, really. We just talked about what the drugs are. We talked about how they work, right? They work through proteins, basically. We talked about the behaviors of the drug, the properties of drugs, the fact that they have to target a specific protein and fit into it, the fact that they create some kind of a biological effect and response from the cells. We talked about the dose, which is very important in terms of what kind of an effect or how much effect you will have from a drug. And then we talked about the genes and how the genes are made. And the link between the two really is the proteins, isn't it? Because the genes are there to produce proteins, and the proteins are targets from drugs. So any change you have in genes, any mutations we might have in genes, that will affect how the protein will function, how the drug target will function. And of course, that will affect the way that the drug is going to affect that protein. I'm going to give you examples of this as we go into the pharmacogenomics. But before I go, I wanted with this slide to highlight the fact that we're all different. But if you think about it, when you have clinical trials and you have drugs or vaccines tested to people in order to see whether it's doing what it's supposed to be doing and whether it's safe and everything, they all give, they give the drugs to all people, same drug, same amount and same dose, right? And of course, you can't do anything else because that's what you want to do. You want to see how the drug affects the general population, and the general population has differences in genes, right? But this means that every drug will have side effects. Every drug. First, because of the action itself of the drug, right? Secondly, because the dose might be not right for everyone. And thirdly, because of the changes and the differences that we all have and we all share through our genes. That creates issues of side effects. So every drug and every vaccine will have side effects to people. The question is, and this is the aim of the clinical trial, to see whether these side effects are important or very more in terms of benefit, and whether it tips the balance towards side effects in response to benefits. If the drug has more benefits, but it does also have some side effects, then the drug goes to the market. But if the drug has a lot of side effects, not so much of a benefit, then it doesn't go. Okay, so before I discuss about the the pharmacogenetics and pharmacogenomics, these are two different words, right? And they're coming from the differences between genomics and genetics, right? Not many people know this, right? Most of the people think they're kind of the same, right? It's about genes, right? It is about genes for both of them. But when you're looking at genetics, you're looking at a particular gene, right? So how this gene affects your life, how it does affect your health, how this particular gene affects how your drugs work, right? This is genetics. Genomics is how many different genes together they affect your health, right? Individually, they might not affect your health. Individually, they might not affect how your body works, but coming together, if they exist together, these genes might give you something different. That's called genomics, right? And the reason why I discuss the difference between genomics and genetics is because if you stick the word pharma next to it, which is basically the Greek word for drug, then you have pharmacogenomics and pharmacogenetics, right? So pharmacogenetics is when you're talking about the relationship between drugs and a particular gene that is doing something, right? That's pharmacogenetics. Pharmacogenomics is when you're talking about a drug with response and in relation to many different mutations of many different genes. I'll give you a short example. Now, let's say that, and I'm going to give more examples as we go and talk about pharmacogenomics and pharmacogenetics, but let's say that I have a mutation in my opioid receptor, right? And I didn't know, obviously, nobody knows about their own mutation without studying them. So let's say I don't know about this mutation, and I take morphine, right? Let's say I need to take morphine. And this mutation that I have affects the opioid receptor, right? In a way that the mutation, the change into the gene code, the change into the protein makes the protein not be able to fit with the drug, right? So if I take morphine, then obviously I'm not going to have the same effect as everyone else, right? Basically, I'm not going to have an effect as everyone else. That's pharmacogenetics, because it looks at this particular gene and this drug, that's it. But if I have a mutation in the opioid receptor, right? But that mutation doesn't really affect a lot the drug. But I also have a mutation in an enzyme that affects the metabolism of morphine. But I also have a mutation in a protein that in my kidneys, and that also affects the absorption, the excretion, sorry, of the morphine in the urine. Now, if you stick all these three mutations together, then that might be a big problem if I take morphine, right? That's pharmacogenomics. So the difference is not huge in terms of a person outside the sciences of drugs, but in the science of drugs is a big difference. Now, unfortunately, pharmacogenomics is sexier in the drug sciences world, so we use it more often, although we want to talk about pharmacogenetics, right? So although we're really interested about a specific relations of one gene with one drug, we don't really call it pharmacogenetics. Sometimes they call it pharmacogenetics, sometimes they call it pharmacogenomics. Unfortunately, people are using these two words interchangeably, and they just mean the same thing, but it's not really the same thing. So I just wanted to clarify this. Now, this is a very interesting picture for me. Now, this is what the pharmacogenetics or pharmacogenomics is all about. Now, let's say that all these people in the middle, right? Let me show you. Let's say all these people in the middle, they are people that have hypertension, blood pressure, high blood pressure, right? And they all go to the same doctor, right? And the same doctor says, okay, you know what? You all have the same high blood pressure, like let's say 50% above normal. So please all take this specific calcium channel inhibitor, right? Now, because all these people have different genes, this drug will be toxic and not beneficial to some people. That's the worst, right? Not only toxic side effects, but also not beneficial, right? The blood pressure doesn't drop. You might have drug not being toxic, but being beneficial. That's the exact group that you want to be in, right? You don't want any side effects and you want the good stuff to happen. But you also have some people that have not toxic, but not beneficial, right? So basically, the drug doesn't do nothing, right? And then you will have toxic drug, so some side effects, but beneficial, right? So the blood pressure will drop, but you will have some nausea, you will have some headaches, right? It might be small side effects, not very important, like nausea or headache might be important side effects. Like it might be very fast beating of the heart, right? It might be excuse me, intense cough like I have now. It might be different stuff. So all these people will be divided into different groups by getting just one drug, all the same drug, right? So obviously in a clinical trial, you expect from the beginning that you will have some people with toxicity, some people that the drug don't work, some people they will work, but they will have some side effects, and then you'll have some people that with no side effects and the drug works. The result is to balance all those four, and measure how many do I have in this group, how many do I have in this group, how many do I have in this group, and then talk about, analyze, and think about whether it's worth it, right? Because if the people that are in the good group are exactly the same as the people that are in the very toxic group, then the drug cannot be used. Because if you use this drug in the general population, you're going to have a lot of people having problems, right? So it's all about the balance. So this brings us actually to the key issue, that is the environment. Now I'm going to talk a little bit about what's called epigenetics, right? There's just a couple of slides, not much, before I go to the core of pharmacogenomics. But epigenetics is not really pharmacogenomics, it's something different, right? The epigenetic science is not looking at how the drug is affected because I have mutations. It's the other way around. It's actually how the environment is causing me mutations, and that actually causes problems into how the drugs are working. So if you see here, our genes are affected by our environment. Our genes are affected by what you eat. Our genes are affected by interactions from the environment, pollution, the amount of radiation, anything that it's in the environment, even the psychological part and the sociological part affects how our genes are working. And this is amazing science, amazing part of science. It's called epigenetics. And we know that if you look at your DNA closely, and then if you unwind it, and you start seeing the double helix, you will start seeing some stuff that are stuck onto the double helix, right? Like you see here, right? And these stuff are attached to specific parts of genes. This is called, for example, DNA methylation, right? Now, what we discovered a few years back is the fact that the genes that have these changes that are happening after you get born, you're born, right? You're not born with these changes. This is what we realized, at least in the beginning. So the genes that have these changes are producing different proteins with mutations, different amount of proteins and different behavior of proteins than genes that don't have these changes, right? And when we start looking at when do you have these changes, it's you realize that you have them in every little bit. Diet produces these changes. The seasons, the weather is producing these changes. Disease is producing changes. The chemicals and the drugs that you use, and the psychology that you have, the exercise that you have, the microbes that you have in your gut, which are good microbes and you need them to digest food, they are creating changes. The every change that you're trying to do, massage or I don't know, any alternative medicine or anything that you're trying to do in your life that affects your body might have an effect on your genes. This is what we call it epigenetic modulation. So it's a modulation, it's a regulation, right? It's a fiddling, it's a change. It's amazing stuff, right? Because we never realized before. We thought that we're born with genes and we die with the same genes, right? And if we have a mutation, we just keep it and that's it. It's the mutation, the hereditary mutations, the ones that we get from birth. But now we know that we have mutations, changes, not mutations, changes in our DNA that are happening because of our lives, right? And guess what? In the few years, a couple of years back, there was a big study that showed that the changes that happened to people because of war in their DNA and because the anxiety and the stress on their bodies because of the war actually was changes that were hereditary to the next generations in 3 generations. So it's amazing science that we discovered that stuff that has happened to our ancestors that they transformed that DNA is actually in our DNA as well. And it affects our proteins and affects the way that we behave as humans, and essentially, but also our body. And if you think about it, that opens up a big part of biomedical sciences, right? Start to think, and actually you can actually use that to explain changes, and you explain traits, and you explain commonalities that you had with other people, etc. And you try to explain things that bond people because they live in the same environment, right? They live in the same environment, they have the same effects on their genes. So this is the science of epigenetics. I'm not going to discuss epigenetics because it's not the focus of this lecture, but I just want to throw it up in there to just give you, open up a small window for you to see that, of course, pharmacogenetics is how the genes affect the drug, but we also have the other way around, right? We have the drugs affecting the genes. That's epigenetics. That's a completely another part of science. Very interesting one. Okay, so pharmacogenomics and pharmacogenetics are looking at the genes and which drug is better. This is the answer that it's trying to understand. Which, if I know that we have mutations, and then we know that we have changes in our genes because of mutations that we carry on, right? That means that I have specific drug targets and specific CYP enzymes that are affecting the drugs, right? So now the question is, if I know your DNA before I give you a drug, maybe I can choose the drug in order to be less toxic and to work better, right? And I'm going to give you examples of pharmacogenetics and pharmacogenomics. This is what it is about. I'm going to personalize the drug that I give to my patients based on their DNA, right? Because some drugs are not going to be effective in this patient because it has specific mutations. Some drugs are going to be exactly perfect for that patient because of that mutation, right? And we have to differentiate that. At the moment, we're not doing it fully, right? At the moment, you go to your doctor, you say, you know what, I have a big headache. They get, okay, just get paracetamol, right? You have a headache, you take paracetamol as well. Everyone takes the same drug. They have the same disease. That's why they take the same drug, right? But they don't have the same genes. What it should happen is that I could look at your DNA. I could see what kind of mutations you have. I can identify these mutations, whether it affects the drug. And if it does, then I can give you a different drug. The one that will be better for you, the one that will be safer for you in order to avert toxicity and also to have better effects. This is what pharmacogenetics is all about. Now, do we do this at the moment? Actually, we are doing it. We started doing it. We don't do it 100%. If you go to your doctor, he doesn't say, look, you know, just give me a little bit of blood. I want to see your genes. Not yet, but this will be the future. It opens up a lot of questions. I'm going to go through them a little bit, but it's the future. And I'm going to give you an example of how we use pharmacogenetics at the moment. Actually, in 2022 that we are now, one part of medicine, which is called oncology, and it's looking at cancer, is using pharmacogenomics more than any other area of medicine. And I'm going to give you examples not only about cancer, but about something else as well. But let's see, first of all, let's see how exactly can we do that? How exactly can we use the genes in order to choose the better drug, right? So remember I talked about the hypertension. Remember I talked about the contraction and the dilation of the blood vessel because of the muscle, and the muscle has calcium that goes in through the channel. So this is the channel, right? And obviously this channel is a protein. And because it's a protein, it comes from a specific gene, which is called calcium channel gene, right? And if you look at the population, all the population, you find that 30% of this population has a specific mutation in a specific codon in a specific part of this protein. That mutation, remember, 30% of the population, that's a huge part, right? These people that has this mutation will have a little bit different shape of this protein than everyone else. Different shape, different effect in terms of drug. Remember, lock and key. If my lock is different, the drug won't work. The key won't work. So then if I know the mutation, and if I know that you have that mutation, then I'm not going to give you drug A, I'm going to give you drug C or drug B or drug D, another drug that it's better and it fits perfectly to the protein that you have that mutation. Okay, that's the pharmacogenomics or pharmacogenetics that has to do with the target, because now we're looking at the target of the drug, right? But we can also look at the CYP enzymes, the liver, because the liver is the one that destroys the drug. So if you have a mutation, look at these people here. These are the general population. Without pharmacogenomics, if they have the same condition, they all take 100 milligrams of a drug, right? But if I look at their genes, I can find differences in their CYP enzymes, and then I can divide them into groups. And I can say, you know what, you have a specific mutation, and you're actually what we call an ultra metabolizer. What's that? Basically, you have a mutation in your CYP enzyme that metabolizes this drug that you take a lot, right? So basically, it means that you need to take more because your liver metabolizes the drug. You can't just take 100 milligrams. You have to take 500 milligrams to be effective. Whereas you guys are fine, maybe you're a little bit less, like 100 milligrams will be fine for you, but you have the poor metabolizers. You guys have a different mutation, and this mutation creates your CYP enzyme not to metabolize a lot, the drug. What does that mean? It means that if I give you 100 milligrams, it won't be metabolized, but I thought it would be metabolized. So I give you 100 milligrams, right? All that will create toxicity because it's a lot of drug. Remember what we said about the Paracelsus? A lot of drug is poison, right? So if I know that you have a mutation, I'm going to give you 10 milligrams, smaller dose. So you see here the difference between before I gave you a different drug, something that is better. Here I'm giving you the same drug, but I'm changing the dose. So your genes actually give me an idea of how much efficiency you will have in terms of the metabolism, and then I can change the dose so it won't be toxic to you and it will be perfect for your condition, right? So as you can see, I can use pharmacogenetics in two ways. First, I can use it in order to choose the drug. Second, I can use it in order to identify the right dose for you. So this is an example. This is a fine example. This is a true example. 5 to 10% of the population, look at that. They have CYP, sorry, it's a mistake here. It's a CYP2D6 deficiency. I'm not talking about mutation. They don't have the enzyme at all. You don't have the gene to make a protein. So basically, if you are a CYP2D6 deficient person, if you try to take these 4 drugs that they need CYP2D6 to be metabolized, you will have very, very high toxicity. You will have unbelievable side effects that might actually endanger your life, right? So this is important stuff, and people that have these deficiencies, unfortunately, they found out through the hard way. They took a drug and they had symptoms, and then their doctor looked at their gene through a genetic analysis, and they found out that they are deficient. And then their doctor say, look, now you're deficient. You shouldn't take this, and this drug. You shouldn't take them, right? So it's very important. And what can this person do? Well, this person can take another drug that is doing a similar job, but it uses a different enzyme to be metabolized, right? So imagine how important it is to know your genes before you take a drug. Because, okay, let's say that you have some side effects, but these side effects are not very important. Okay, let's say that you have a little bit of headache, I had a little bit of fever, but then it went away. That's fine. But you might have a very, very high toxic effect, which might endanger your life. And then that might be very, very important to know beforehand. So in pharmacogenetics, in pharmacogenomics, what we are doing now, or we used to be doing a lot just a few years back, we didn't have pharmacogenomics. So what we did, we gave the drug to everyone, same drug, same dose. Then someone died or someone had side effect. And I thought, you know what? These people that have these problems, a small amount, but they have problems. Let's look at their genes. And then we found out about the polymorphins, the mutations that these people had. But that's not the right way to do it, because people are dying and people are having side effects, right? What you should be doing is using these analyses before you give the drug to people. So first you're doing this analysis, you identify people that have this mutation, and then you don't give that drug to that person. So you save people's lives and you save side effects. And just to be clear about this word that you see here called genotype, and then you see another word here called phenotype. Genotype is your gene profile, basically all your genes, right? If I know them and I have them in one page, that's your genotype, right? Phenotype is what you look like, right? What characteristics that you have, right? And the genotype and the phenotype are two important things, of course, in pharmacogenomics, right? So let's say that you want to use pharmacogenetics, but you want to use it in a clinical trial. All right, let's say that you discover a new drug and you want to cure people with this new drug, but you want to know a little bit about the pharmacogenetics of this drug. One way to do a clinical trial is this. So, for the people that, of course, that don't have a background, a clinical trial is when I test a drug into many people, right? Remember when the vaccines needed to come out for COVID-19, we were waiting for the clinical trials, right? We're waiting for the first people to take the vaccine to see what it does. So in the clinical trials, a standard clinical trial is called a randomized clinical trial, RCT. The word randomized is very important. Why? Because I have to give to compare people that take the drug and people that take a placebo. So how do I divide these people, right? I don't pick them, no, because that will be biased, right? It will be subjective, right? It's not objective. So what I'm doing is I'm randomly, actually we use computers for that. The computer is randomly dividing the people, and it divides them to a drug A and to two groups, and in one group we give the drug, and in one we don't give the drug, and then we're just waiting to see the effect, right? This is a standard clinical trial, right? But then the people that took the drug, we genotype them, right? We look at their genes, and then we define their genes in terms of mutations. We split them in groups depending on whether they have a mutation or not. And then we compare the effect between those little groups. I'll give you an example. Let's say that we are in a clinical trial for a vaccine, right? And I am randomized by the computer. I join a clinical trial. I'm randomized by computer, and I get into a group that I'm taking the vaccine, right? And I take the vaccine, and then the doctors are waiting to see whether I'm going to be infected or whether I'm going to have an effect or something. And let's say that we are like 10 people that we took the vaccine, and we wait, and okay, they measure the effect, whatever the effect might be, and then the clinical trial finishes, right? Then they say, hang on a minute, you 10 guys, give us your DNA, let's check your DNA. And then let's say that I have a mutation in a specific protein that's important for the vaccine, right? They put me in a different group, and then they look at the effect that I had from the vaccine just from that group, right? This is important because it will identify whether my effect, the effect that I had, was because of a specific mutation that I had in my gene or not. And of course, the same happens to the people in the placebo group. So this is genotyping after we get the treatment, right? But we can do genotyping before actually the treatment as well. So in another design of the trial, first we do the genotype. We divide all the people between the ones that have mutation and the ones that don't have mutation. And then the ones that have a mutation, these are the ones that we split them into two groups, a placebo and a drug. Why? Because in this way, all your people will have the mutation. You won't have an unknown person with a different genetic background to mess with your data. So basically, you study the people with just this mutation. This is called genotyping before treatment. Okay, perfect. So we finished with the different ways of how the drugs, how genotyping can work. Now let's go and check a few examples, and then I'm going to talk very, very shortly about the future of pharmacogenomics, and then we're done. Examples, right? Look at this, breast cancer, right? Very difficult disease, but thankfully we have a lot of drugs for breast cancer, which are very good drugs, right? Plus we have surgery. So breast cancer is no longer a very huge life-threatening condition. It's actually treatable. And we have a very good drug called tamoxifen, right? For breast cancer. And tamoxifen is metabolized by CYP2D6, right? But the metabolite, which is a change drug, which is called endoxifen, this is actually the one that has the good anti-cancer properties. Okay, so this is a little bit strange. The drug itself doesn't kill cancer, but once it goes into your liver and your liver changes it to endoxifen, the endoxifen has the anti-cancer effect. That's important. Why? Because people with normal CYP2D6, fine, it will work fine. Why? Because tamoxifen will change into endoxifen and then your cancer will be killed. People without CYP2D6, they won't be able to have any treatment from endoxifen because they won't have endoxifen, right? So the drug will be useless. And people with a mutated CYP2D6 that might have high metabolism of tamoxifen, that means a lot of endoxifen produced, that will kill cancer, okay, but it will also have a lot of toxic side effects, right? So you see here that in real life, in breast cancer, it's very important before a person with breast cancer gets tamoxifen to do a genotype analysis to see whether you have a CYP2D6 change. Another drug for HIV, abacavir, right? This is a very good antiviral drug against HIV. But apart from going to the virus itself, it goes also to another protein called HLA-B. That's our protein, right? And if it goes to a normal HLA-B, then it's fine. It doesn't do anything. But if you have a mutation in that protein, you have a lot of side effects, right? You have intense nausea, diarrhea, fever, skin rashes, etc. Right? So there is a recommendation. Your doctor requires to take a genotype from you before he gives you abacavir. He wants to see whether you have that mutation of HLA-B in order to avoid giving you the drug whether you have that mutation or not. These are all real-life examples that we use in the clinic for pharmacogenomics. High cholesterol, right? I'm sure your grandpa or your grandma or someone very close to you have high cholesterol. Very usual condition, right? And we have some drugs that are called statins. Basically, any drug ends with statin is a drug that reduces cholesterol, right? And we have more than 10 drugs, right? We have a lot of drugs to choose from. They're all doing the same thing. But their target, which is a protein called HMG-CoR protein, 80% of the population are fine with it, but 20% of the population will have a mutation. And then specific drugs won't be effective, right? And you might say, you know what, fine, if it's not effective, what's okay, if it's not effective, the doctor will see that it's not effective because my cholesterol won't go down and it will give me a new drug. Why do I need to do genotyping? Well, remember, the people that have high cholesterol, they have high risk of heart attack, right? Because the cholesterol will block their arteries and then they have a heart attack. So if you have a high cholesterol and you take a drug, you don't want to risk not working, right? Because you take the drug and you think it's working, you find out that it's not working some weeks or months afterwards. And then in the meantime, you'll have a very high risk of heart attack. So it's very important to know whether you have a mutation, because if you do know, then you will avoid a certain drug and you can pick the best drug out of these 10 drugs to have a better effect. I have other examples as well by other drugs for breast cancer. I'm going to pass it through a little bit. Melanoma as well, another cancer that requires, we have a specific drug. This is kind of interesting. So here is a drug that is used for melanoma, for cancer, right? Skin cancer, basically. And this drug requires you to have a mutation in order to be working, right? If you don't have a mutation, if you have just normal, the wild type protein, it won't do anything, right? So it's recommended by the health organizations and the regulatory organizations. It is recommended that you do a test before you take this drug, right? And only if you do have a mutation, then you can take this drug. Okay, so now let's talk a little bit about the future, a couple of slides, and then we're done. I gave you an idea of how drugs work and how drugs do what they do through their proteins. And then we talked about the genes, and then I gave you an idea of how the pharmacogenomics can be used in real life to avert you not only from toxicity and risk of death, but also to give you a better chance for a better drugs. And actually, this is happening at the moment, not fully, right? In 2014, many years ago, but I'm sorry, this is the date that I have the data from FDA. 41 drugs were approved, new drugs were approved in 2014. 8 of them required a genetic test before you take them. And in Europe, which is the EMA, is the same body as FDA in the America. In 2010, look at this here. This is 2009, 10. This is the number of new drugs, right? Every year approved, right? Number 10, 11, 2012, 2013, 14, 15. Every year, we have more drugs that have genomic biomarkers. Genomic biomarker is exactly this. It requires you to check for something in your genes before you take the drug, right? So every year, today we have 2022, we have a lot of drugs that are coming out that require you gene testing beforehand. And now think about epigenetics as well, right? All these things in our life that affect our genes. And you have the genetics, and you have the pharmacogenetics and the pharmacogenomics, and then you have the analysis and the technology that we can use in order to bring those two together. So the future of pharmacogenomics is seen by people even more wise than me in terms of how drugs are used. They are looking at exactly this. I don't know if it's going to be in 10 years or 20 years or 30 years or something, but we are going to be needing a gene test, and we are going to have our gene profile in a chip or something to give it to the doctor to check it before he gives us a new drug. That's the future of drug science. And then if we do that, we won't have any side effects of any drug, or we will have the minimum possible, right? And this is what you want to do. Imagine a world where no matter what kind of drug you take, what kind of disease you have, no matter what vaccine you take, you won't have any side effects, right? Imagine if the vaccines that we had today in this COVID pandemic had 0 side effects, right? Imagine the drugs for cancer, which are very toxic, had 0 side effects, right? It would be a better world, definitely. And it would really help us understand how to best use that. So I'm going to give you, finalizing, an idea of how is this happening now. You have companies now, companies that they are asking for they are doing gene testing for you, right? They send you a kit, you take a little bit of you swap it in your mouth to take a little bit of DNA, you put it in a vial, you send it to them. They, of course, you pay them, right? And they analyze your gene on specific drug targets for specific drugs. And then they give you a feedback and they give you ideas. These are links. Just to be clear, I have no interest in any of these companies and I don't have any links with these companies. I just Googled them and you can Google them and these are the first ones that came out. Gene testing, if I click that, I'm going to open my browser, just 1 sec. I want to give you an idea of what this looks like. And it's very interesting, actually. So you open up these companies called GeneLex, right? And it gives you a full menu. If it opens up in a minute, it will give you a full menu of, yes, there you go. So it's GeneLex pharmacogenomics testing, right? It tells you a little bit about pharmacogenetics and what it is. And look at this here, the test panel. So these are all the genes they will look for you. Look at that, all the CYP enzymes here, some other enzymes. You have some drug targets. There are not many, okay? This particular company is doing only 30, but if we look at every one of these genes, for example, this gene here, OPRM1, is the gene to create the morphine receptor, right? So if I click it, I think it gives you a little bit of details. Yes, so look at that, OPRM testing. These are the opioid receptor, right? And it tells you about the receptor and it tells you about what kind of drugs are being used for this receptor. So all these people are doing is they send you a swab and you do the swabbing, and then you might ask you for blood, depending on what you want to do. And then in 10 days, they give you the test, the results back, right? So I'm not going to open up all the others, but all the others are exactly the same. And this is what is being done. Now, of course, some of you guys are maybe into law or maybe into ethics, but as you probably understand, of course, this is the future, but it's very, very tricky, right? Because now imagine we need to be showing our DNA profile for every therapeutic intervention, for every drug, right? It requires it has some ethical issues and considerations, right? We need to regulate these companies. These companies are just doing testing, but we don't know. Are they keeping the DNA that we sent them? Do they put it in some sort of a database? How long do they keep it, right? Do they throw it away? Who is looking after our ethical interests, right? Who is overwatching for quality, right? If I send the DNA to them, and if they give me back the data, can I trust the data? Can I trust that they did a good job? Because I'm going to be basing my clinical intervention. I'm going to be choosing which drug I'm going to take. This is my life we're talking about, right? Based on that. So we really need to be talking in the next decades or years about quality assurance and overwatch. And these are all the questions which I was able to find in the literature about cost and benefits, because if genotyping is very expensive, then it doesn't worth it, right? And we need to be looking at commercialization and logistics and ownership and ethics and expertise and all that, because let's think about it. Does your doctor know about pharmacogenetics, right? I don't know. Not many doctors know about it. I mean, they've heard about it, but they're not experts. So if you give your gene profile to your doctor now or tomorrow, will they know what to do with it? I doubt it, right? So I'm working at a medical school and we're trying to teach our students now, the future generation of doctors, how to use pharmacogenomics based on the data that we have. And this is where the future will go, both in training, education, but also in terms of patients and drug science. And that's it. Basically, I have a couple of links here as resources, and I'm going to be, I'm happy to give you actually all the slides. You can download them by using your phone to just click on at that. Basically, if you use that class code and take a picture of that, it will pop up a website, and you just add your name, and then you can download the slide by using the download button from that class. So I'm going to leave it here, and I can get actually questions. Let me just put it down. So you don't have to put your own normal name. You can put a nickname if you want. It's anonymous completely, but you can download that. I'm going to keep it for now. Let me see. Actually, I can give you the, yes, how are you going to know though? Give me 1 second. Let me just do it again once more. Right. So can you guys can see it, right? Yes, you can see it. I'm sure you can see it. Yeah. Thank you very much. Okay, perfect. So that's it basically. Of course, I'm gonna download and I'm not gonna upload this lecture and these slides to the YUFE website. I think there's a specific place where you can do that. But I would think I can also send it to you as a PDF if you want, through emailing, but it might take a couple of days. Now let's see. If you guys have any questions, I'm very happy to answer them. Let's give a couple of minutes to people that might need to answer some questions. No? Okay, perfect. Let me see the chat, because we do have a couple of, thank you for the lecture. Okay, perfect. Thank you. Will you share the recording with us? Okay, perfect. Thank you very much, Andrea. That's a very good question. If the YUFE website allows me to upload the recording, I will. If they don't, I'm going to have to ask the university whether I can share the recording, because it has some names in there and some personal information. So I want to make sure that we can do that. So I'll ask if we can, and if we can, and if I can upload on YUFE, you'll know. If I cannot upload on YUFE, I'm going to send the recording for you to download if I'm allowed to. And I have another question. Wondering if you could have access to the presentation. Okay, that's fine. I'm interested about more for breast cancer drugs, which you skipped. Okay, right. So the drugs that are skipped are just examples, right? You're gonna be able to look at the presentation. I just wanted to be a little bit quicker. But there are examples. Basically, we have many examples in the links of the webs of this presentation. I have a couple of links at the end, right? If you click on that link of the FDA, it's actually here. But if you click on that, once I give you the presentation, you will see all the drugs that are required for a cancer, that are requiring to do genotyping. All the drugs, even for non-cancer, for everything, right? So we do have nice resources for that. Let me see if I have any other questions on the chat. Thank you very much, Killian. Thank you very much, Nafsika. Thank you very much, Lucas, Marcos, and everyone. You're very kind words. If anyone wants to talk and ask a question orally, you can unmute yourselves. I think you can. Yes, you can. And you can just ask for just a couple of more minutes. All right. Thank you. I don't think I have any other questions here. I'm going to keep it here just a couple of more minutes open for people to snap, to take a picture of this if they needed to. If anyone else wants to go, thank you very much for attending this lecture. And I wish you all the best for your studies. See you later, guys.
Thank you. Have a nice day ahead.
Thank you very much. Goodbye.
Bye.